Hey everyone, I'm going to be giving an astrobiology talk today. This one's about one of the two main origins of life hypotheses. This is abiogenesis, where chemistry became biology. There's two main thoughts, and one of them was actually discussed in detail on another channel called GeoGirl, which is a great channel you should check out. That one talked about the hydrothermal vent hypothesis, and this chat's going to be talking about the warm little ponds hypothesis for the origin of life. And this is actually based off of reading this book by David Deamer. You might know his work. He's a wonderful scientist. This book is worth checking out if you're interested. It's not that long. Actually, someone else I know thoroughly enjoyed this and actually used it to inform a textbook that they ended up writing, which is very cool. And I've kind of given this talk. It's a few years old now. I gave it for a class and a few lectures that I've taught myself in front of university students. I've kind of given similar talks and also gave this talk at a symposium. But anyways, kind of getting a little ahead of myself. So let's just start. So stromatolites, which you've probably heard of, you can see them right here. You can see some actual modern photos, but we also have fossilized stromatolites, and they can tell us about depositional environments and whatnot. But they're first discovered in the Pilbara region in Western Australia, and they're just microbial mats that become mineralized as they grow on rock surfaces. You can almost even think of this as like plaque growing on your teeth, like before you brush your teeth, if there's a buildup of stuff that you can kind of like scratch off. This is kind of just minerals, and various things kind of get caught, and these microbial mats just basically grow in layers. And this is believed to be the first form of life on Earth. So on early Earth, there's an ocean with volcanoes. We've passed the late heavy bombardment. The surface is cooled. The moon origin event with collision with Theia that created the moon, that's all happened. The Hadean period's ending, Earth is cooling down, and now we have oceans formed. We have escaped water vapor from cooling, melting rocks. We know from stable isotopes that the Earth's water was partially delivered by comets, not entirely, but also just water that was trapped in the planetesimals that formed Earth. And then as Earth cooled, they condensed and precipitated out, and we have oceans. Stable isotopes of hydrogen, looking at deuterium, so that Earth's water is mainly delivered from the planetesimals that actually built Earth, but meteorites from the asteroid belt have similar deuterium-hydrogen ratios, so that they likely have a similar origin. It's one of the beautiful things you can use stable isotopes for, tracing and thermometry, and this is an example of tracing. And we know that these materials also delivered organic compounds, metals, carbon, oxygen, nitrogen, potassium, sulfur, things needed for life, all the big elements. We also have zircons, which have liquid water in an atmosphere. The zircons were dated to 4.404 billion years ago. The G stands for giga, like gigawatts or gigabytes. So we know that at that time, we had liquid water in an atmosphere on Earth from dating of rocks that existed at this time. There's not many rocks that have been around there. Terrestrial rocks are, in some areas, older than ocean rocks. But, yeah, so we have an idea of this. So Earth, 4 billion years ago, again, from these kind of paleoclimate archives, which I'll talk about in another video, and I have in some of my shorts here on this channel, show that the average temperature was maybe about 55 to 85 degrees Celsius. This provides activation energy. Ocean pH was probably about 5 or 6, so just on the acidic side. And we had low atmospheric oxygen because the Earth was low in oxygen early in its life history, and it wasn't until cyanobacteria and life later, and if you're interested, I can do lecture on the great oxidation event, created the CO2 that we now need and breathe and whatnot. The sodium concentration was probably 1.5 to 2 times more than it is in the early ocean. You can look at an oxygen isotope study, Noth et al., or sorry, just Noth 2005, talk about this in more detail. and basically just dissolve soluble ions from minerals within the crust, go into the ocean, and make it even saltier than it is now. No cells that we know of function with high concentration of sodium chloride. They actively pump out sodium chloride in favor of potassium, and this is one of the main ionic species in hydrothermal systems. So perhaps this makes us think, okay, maybe there's potential connection here. Why is low pH needed? Well, it dissolves apatite and it releases calcium and phosphate into solution, which can then be used up. This actually solves what's known as the phosphate problem, which you can look up or I can talk about in more detail. And it's also needed for polymerization. And phosphate is essential for ATP. It's a linking molecule in a polymer structure of nucleic acids. And we also have enzyme-catalyzed ATP hydrolysis, which is needed for maintaining ion concentration gradients within a cell. This is just an experiment that was done. They had pure seawater. They have seawater with apatite. And then if you bubble CO2, which decreases the pH, you can actually get rid of that apatite and you can release all this phosphate. So something similar may have happened in early Earth, which allowed this phosphate to be available. Early Earth lacked continents at this point in time and was covered by a salty ocean, likely several kilometers deep. But little by little, volcanic islands began to form, resembling Hawaii and, let's say, Iceland today, or similar to Apollinaris Mons on Mars, which was also geologically active and alive roughly 4 billion years ago. You can see early Earth 4 billion years ago on the left, and early Mars 4 billion years ago. It was warm and wet and had an ocean and could have potentially caused life, or had life there too, which is very cool. And I've done some work with NASA that explores that I can, again, maybe talk about in another video. So hydrothermal conditions are conducive to the origin of life. This is a photo that was in Astrobiology magazine taken from Yellowstone National Park that shows a hydrothermal vent pool, which we'll talk about earlier, but this is where that chemistry to biology environment may have happened. So volcanoes on early Earth were highly active with eruptions of ash and lava. They were the first land masses. Volcanism produced hydrothermal vents. However, the absence of continents and plate tectonics made them look like seamounds over large magma plumes. Evaporation of water from surrounding seas can then fall on precipitation of the flanks of these volcanoes that have now peaked above the ocean and formed the first land masses. They can flow downhill and they can accumulate in freshwater hydrothermal fields, making these warm little ponds. The hydrothermal rocks themselves are basalts, right, that cooled lava. Eventually can be altered to form clays, and after millions of years of exposure to acidic water, due to hydrochloric acid, HCl, and sulfur compounds, and elevated temperatures, they can begin to create aluminum phyllosilicates. This is an image from the David Deamer book. So you have these volcanoes that are starting to poke out, kind of like happens Hawaii. Right? You have they erupt. There's ash. There's hydrochloric acid, sulfur dioxide, carbon dioxide, water vapor falls down as acid rain ash. And acid rain and ash. There's pyroclastic flow. There's mud flows. There's lava, and then as rain comes, you get these little pools of water that form, and then they can start to alter the basalts and produce clay minerals, which I'll talk about why that's important. So clays themselves have large surface area and are negatively charged, which has a positive effect. They can adsorb organic solutes, which can become concentrated on mineral surfaces, which allows interesting chemistry to happen. But there's a negative effect is the adsorptive capacity can also remove solutes from a solution as active reactants, and basically once they get stuck to the clays, it's hard for them to get unstuck, and they might just be permanently removed from the system. So there's no guarantee that they'll undergo useful reactions if they are bound to mineral surfaces or are no longer freely diffusing in the environment. This is just a schematic of clays. We have kaolin. There's illite with these, you can see the K plus layers here. And then we have bentonites, which is just weathered volcanic ash, which is really cool. You can use that for dating as well. You can do like potassium argon dating. Just as an aside, you can do stuff like the Cretaceous and kind of date periods and stuff like that as well. But, a bentonite, or like even like a smectite, they're known as swelling clays because they can actually trap water and expand or swell. This is a picture of montmorillonite and kaolinite, just to give you a picture of what some of these clays look like under an electron microscope. And we also have these hydrothermal fields, these ponds with clay that's accumulating. We have Mount Lassen and Mount Mutinovsky that actually show similar environments, which is cool. So we know these processes are happening, and we know that interesting things can happen from here. So let's look at sources of organic compounds required for primitive life. And again, the talk was focusing on the book, so I haven't adapted too many photos from outside of that, but again, I can talk about that in a future video. So there's multiple sources of biologically relevant organic molecules. There's interplanetary dust particles, IDPs, that are falling consistently. They introduce volatile compounds into the atmosphere. There's also the atmospheric and geochemical synthesis of compounds like formaldehyde and HCN, which react with amino acids, nucleobases, and sugars. We observe this all the time. During this period, there's an absence of oxygen on Earth, like I mentioned, so there's no oxidation of these organic compounds. They can stick around. We have the ESA Rosetta mission. Basically found that all these different compounds are found in interstellar space. They're brought to us not just from interplanetary dust particles, but we found that they're present in asteroids. Even like asteroid Bennu or Ryugu have these compounds. Multiple different meteorites that have landed on Earth, we see them, even comets and whatnot. Mineral particles collected from cometary dust stream in 2003, when the Earth passed through the tail of a comet. Also found similar data. Particle and organic material on the surface are likely to be left over from the molecular dust cloud from which the solar system formed. And this is just a comparative plot of some of the major soluble organic compound abundances in the Murchison and CR2 meteorites, which also has a lot of amino acids, which are used to make proteins and needed for life. Or we can find of the 20 amino acids, 10 have been, that's the Miller-Urey experiment. I'm getting ahead of myself. But amino acids have been found in astromaterials, in extraterrestrial material. Origin of key molecules, won't go into this, but Strecker synthesis can create amino acids. Formose reaction can create carbohydrates. Fischer-Tropsch type synthesis can create amphiphilic compounds. And then we have concentrations by clays. And we have this study from 1990 that showed that synthesis of biologically relevant monomers can be driven by interactions with mineral surfaces. And this is just an example of the photochemical synthesis of formaldehyde, which is just H2 and CO2 reacting with UV from the sun to create HCHO, then HCHO, and then fall as rain and be on Earth's surface. And again, we've observed this in hydrothermal pools. The circulating water in hydrothermal fields was moderately acidic and had a pH of 2 to 5. They can have concentrations of common cations such as potassium, calcium, and magnesium, which we've measured, accompanied by anions of silicate, sulfate, and chloride. And then we have what are known as wet-dry cycling. So wet-dry cycling is actually really cool. And this has actually been studied in the lab too, and you can check out a bunch of papers about this. But basically, you have an inland hydrothermal site subject to cycling. So you have water that has fallen from rain, other molecules and things that have concentrated, like formaldehyde and whatnot. And they basically go through periods where there's a pool, and then the water evaporates, and that's the drying phase, right? So the wet-dry cycle is when there is water in this area, it's wet, and then when the water evaporates, it's dry, and these molecules basically stay. You can see here there's a hydration phase where the phospholipids that are in there, the fatty acids that can also form, can actually trap material and form a layer which can contain and concentrate materials during the wet phase, and then when they dry, they kind of flatten out and form these layers during the dehydration or dry phase. And during this time, polymerization can occur, and then when they rehydrate, you can get more complex molecules in the new little bubbles that have formed. And then over time, you can get more complex chemistries like that. And again, this has been studied pretty in depth. So again, you have multiple cycles, and then over time, you can get some things rupture and introduce them back into the layers to be used for other cycles, and then you have other ones that can continue to stay sealed. I should have had that there. If organic compounds landed in the ocean, they're automatically diluted and they can't do anything interesting. But compounds landing on or synthesized in volcanic landmasses could be exposed to the atmosphere. They would become concentrated on mineral surfaces. There is UV sterilization, which is by water. There's precipitation, which can flush out compounds into hydrothermal pools, which can concentrate them. And there's a turnover process. Basically, they're delivered, they're synthesized, they can be degraded, new molecules can be recycled into other processes, etc. The main components of life can be chemically synthesized in the lab. These materials are also present in carbonaceous chondrites, and 10 amino acids found in meteorites are among the 20 amino acids that are used for life. We've found them multiple times. There was another study that was done that actually said that from those 10 that you have, you can potentially get the other 10. So how do we sort out these organics? How do we concentrate them? And how can prebiotic chemistry be maintained in which synthesis and decomposition rates are balanced? Well, there's a process of self-assembly. So rare polymers become functional structures if they are encapsulated and closed in these membranous compartments that I kind of showed, those little bubbles that I mentioned, and subjected to selective processes. So lipids that's what forms the outer layer of these bubbles, and I'll tell you their properties soon. But they're stable due to the hydrophobic effect. So they originally come together through van der Waals interactions. Entropy increases if the hydrocarbon chains are together, and they originally form a micelle, which is a single layer bubble, then a vesicle, which is a double layer vessel, which then during the drying phase can become a multilamellar structure. Those are those sheets during the dry or dehydration phase. Proteins can become hydrated, proteins provide hydrated channel through the bilayer, and you can actually have electron and proton transport during this time. This is a picture here. So these are those bubbles that I showed in the other figure. So you have, this is water, there's a monolayer of these lipids. They can come together and form a micelle. So you have, let's say, the heads on the outside and the tails are on the inside, and they're hydrophobic. Lipids are, just think of them as like fats. And then you can actually have these more complex double layer structures called vesicles. So these are the bubbles that I mentioned earlier. Or basically you have the heads here, tails, and then there's another layer where the tails of the outer layer touching the tails of the inner layer, and then you have the heads out there. And then during the drying phase, you get these multilamellar sheets that can trap things and have polymerization. And then you have the mineral surface at the bottom, which can help and concentrate stuff as well, right here. So vesicle formation during the hydration phase, you can see here, when it's wet, these multilamellar phases start to kind of bud off and form these bubbles and these vesicles. And you can see that some of them have stuff in them, some of them are empty. This is an image of this happening actually in the lab. They're purposely set to glow so they can be observed. And then, yeah, so you have some of this material here. and then it buds off and then makes a vesicle. So these membranes of the vesicle require moderately acidic 3 to 5 pH fresh water, which we had, relatively low concentration of divalent cations, composed of chains of 10 carbons or longer, and nonpolar compounds, such as PAHs to promote stability, polycyclic aromatic hydrocarbons or PAHs. And then you can look at this Black and Blosser 2016 paper that looked at adenine can stabilize fragile fatty acid membranes against salt. Again, these fatty acid membranes, these lipids, the membranes of the vesicles. And then again, you can see the survival and disruption of these vesicles, which can eventually start to become a protocell. So if they break, they fail, this material goes back into the layer and can be used by something else. Some of them never captured anything, and some of them can have interesting chemistry that can then, during the dry phase, incorporate more materials like stuff that has come out. Some of these materials can also help reinforce the stability of the vesicle and make it more likely to continue, and as things become more complex, they can start to become protocells and then potentially one day cells. So condensation reactions can actually synthesize random polymers, right? All this material that's fallen is completely random and caught between the phospholipid bilayers and concentrated within vesicles. Condensation reactions, they're defined as chemical reaction in which elements of water are removed between two molecules and they end up forming a linking bond. This can synthesize biopolymers such as nucleic acids and proteins, which are used in life. And polymer synthesis are synthesized by concentration reactions actually occur faster when they are hydrolyzed. During the dry phase, right, if amphiphilic compounds were present, self-assembly of organized multilamellar structures occur, and we've observed this in the lab, we know this well, and these layers were form films. They have liquid crystals in which solutes dissolve and diffuse and are concentrated and can organize into more complex structures, which again we've observed in the lab. Reactions occur between bilayers of multilamellar structures formed by these amphiphilic lipid compounds, and reactions are basically driven by low water activity, since it's the dehydration phase. This is where all the action happens in the anhydrous phase or the dehydration phase, and they could be linked by ester and peptide bonds. And the anhydrous lamellae, those layers, those amphiphilic compound layers, can concentrate monomers, then organize them in such a way that polymerization is actually promoted, and again, more complex things happen. So upon rehydration, these polymers are encapsulated in these microscopic membranous vesicles. Each vesicle is unique, has combinational chemistry. Certain combinations of organic molecules in vesicles will be sufficiently concentrated to undergo chemical reactions and create primitive metabolic pathways. And again, they capture chemical energy and transform potential nutrients. And most energy driving metabolism in the biosphere is supplied by sunlight coupled to photosynthesis, and has similar kind of things going on in life now. So once again, we have the dehydration phase. Things are concentrated, polymerized, they're rehydrated, form these vesicles, right? They bud off, then water evaporates, they begin to form this gel, and then full evaporation, they form these layers, and more complex chemistry can happen. And during the rehydration phase, vesicles burst. The ones that are more robust survive, and the ones that burst provide new material or they can wash away and bring new material, because when they get wet, it's through rain and whatever. They can bring new material, new vesicles that have survived in other environments, etc., further complicating the environment. And all this doesn't happen overnight, right? This takes time. This is not something that takes place even over hundreds or even thousands of years. We're talking millions to even billions of years. Pre-metabolic chemical reactions in hydrothermal conditions. So Martin and Russell 2007 and Sojo et al. 2016 showed a reduction of CO2, and vent conditions might be a source of reduced carbon compounds. which support primitive metabolism. This was tested by Lane in 2015 and Jackson 2016, which was a skeptical paper. But all these papers are peer-reviewed, and people are constantly trying to debunk it, and the best minds in science can't. Mineral membranes are much too thick to function as chemiosomatic membranes because chemiosmosis requires a thin membrane to harvest energy of a pH gradient. Primitive metabolism must function in compartments, so chemical energy is then generated in a closed membrane bounded compartment, and if electron donor at a higher redox state is present in the external medium and the acceptor at a lower redox state is inside, then that helps. If a carrier that can partition into a lipid bilayer is present, then is able to pick up electrons from the donor and transport them across a membrane to the acceptor. So we're seeing more complexity, more evolution, and again, closer to a protocell and a physical cell. Proton gradients can actually be produced across the boundary membranes of vesicular compartments. Sulfate and CO2, which is very present in these volcanic areas, have low reactivity. They're unlikely to be electron acceptors, but iron readily accepts electrons, either as ions or compounds, and can play a major role in electron transportation like they do today. And we also have reduced sulfur that can act as an electron donor. Sources of redox energy are also very present in these environments. So we have sunlight, there's photochemistry, we have sulfate, bicarbonate, carbon dioxide that's forming. There's a redox boundary in the sediment, and then you can have reduced sediment as things get buried. And again, we have the water basalt reaction creating clays that can concentrate things potentially. There's the rock substrate, there's magmatic heat sources, which can provide sources of reducing power. The areas that are buried have no oxygen, so they could reduce and create like FeS2. So the deep interior of the Earth, it's a great source of reducing power in the form of gases like H2 and CH4, right? Hydrogen gas and methane, and they're produced by reactions of water with minerals at high temperatures and pressures. And primitive life could easily take advantage of this available energy. So future research, again, this is an old stuff, so some of this research is going to be done, but just looking at how membranous compartments could have captured light energy using a plausible pigment system. You can look at activated phosphate could be generated in plausible prebiotic conditions. A lot of different work. And then, again, this is, I think, where this gets really interesting. So we have these volcanic islands. There's water, there's material. We talked about some of the chemistry of how that can happen. And different pools can have different conditions, right? So it rains, water collects, wet-dry cycling, etc. forms these vesicles that can become more robust. Maybe because the ones that aren't suited for that environment break down and are destroyed, right? It's natural selection, just like happens with organisms. Can same thing happen with these vesicles. So if you're in a pool that is exposed to high temperature, if the vesicle is not conducive to survive in high temperatures, it won't. But the ones that do survive through natural selection, and again, multiple generations, you can think of the wet-dry cycling as creating multiple generations, can actually create the vesicles that can survive those. And then if that pool gets flooded or there's runoff, it can bring into another pool, and now the ones that survive the high temperatures are now exposed to, let's say, ones that have higher salt. So then some of those ones that can survive are now able to survive high salt, high sodium, and high temperatures, right? And then they can continue to be pushed, and over time you can start to get these really robust cells, and then as you get close to the marine system, you can maybe start to form protocells, cells, and then things that eventually form stromatolites, which links back to the beginning of this lecture. So the functions of a living cell therefore emerge first by chance, followed by selection and refinement, and then gradual incorporation into increasingly functional protocells which each cycle. And this is a graphic that shows it. We have the interstellar medium, which transports stuff to Earth. It gets concentrated and modified and improved. It accumulates, it concentrates. You have the origin cycle of this wet-dry cycling, which can take place over a very long period of time. Then you have distribution, right? The protocells, the vesicles that have survived, let's say, high temperature, now high pH, high salinity, etc., now spread, and they become even more robust and able to survive all of those conditions. And then over time, they can adapt further, and then eventually they can colonize and form life. And this isn't to deny God or anything like that, too. This, you can, if you want to take the religious route, this could just be the way that God works. I don't want to get into that argument, but it could just basically be a likely way that life may have formed. And then there's multiple experiments that have tried simulating the prebiotic environment. There's the Miller-Urey experiment, which I mentioned earlier. It's very famous. Expose a mixture of hydrogen, methane, ammonia, water vapor at atmospheric pressure, and a spark of discharge. The discharge chemically activated the gases and compounds like HCN and HCNO, which we've talked about earlier in this lecture, and they were found to form amino acids. They didn't exist before, and then they existed then. And they found other compounds, which were formed through Strecker synthesis. And then there's other studies. They automated cycling to expose glycine solutions, show that even the simplest process parameters can have high conversion of amino acids, monomers to peptide oligomers, which can become basically more complicated. And basically that's what a lot of these different studies are showing, or things are basically supporting these kind of arguments, and we're actually seeing stuff in the lab. And there's actually more examples than when this presentation was done, but I kind of just liked it and thought this was an interesting thing. Ferris et al. 2006 incubated chemically activated ribonucleotides with montmorillonite clays and monomers absorbed to specific sites, and found that, again, montmorillonite is like the bentonite that I mentioned, can be used for the weathering of volcanic ash, and clay can be considered to catalyze the reaction when monomers are assembled, because the stuff sticks to it and things encounter each other and react, and because they're concentrated, and again, you start forming this interesting chemistry. Rajamani et al. 2008 and found RNA-like polymers which can synthesize non-enzymatically from mononucleotides in lipid environments. So again, you can have RNA, which came before DNA, right? Ribonucleic acid before deoxyribonucleic acid. They're able to form without an enzyme to actually help catalyze this process. So a lot of increasingly cool stuff. And then you can look at gel electrophoresis analysis, revealed that RNA-like polymers were synthesized from mononucleotides by wet-dry cycling. So again, this is strong evidence. De Guzman exposed mononucleotides to multiple wet-dry cycling in the presence of lipids. Used AMP and UMP, and just basically introduced, or when energy was introduced, when water, sorry, my phone went off, and if you heard the Back to the Future chime there. Energy introduced when water activity decreased, ester bonds formed. This linked monomers into polymers, then into oligomers, which then accumulated in kinetic traps, and then you have this kind of hill climbing, which was continued to increase by cycling. And they used CO2 to maintain an oxygen-free environment to protect from oxidation damage, which would have happened in early Earth. And this is just their setup. And again, there's a lot of different things that need to happen. It is complex. And then we have testing in prebiotic analog sites, which can be done, and anyways, you kind of get the idea, right? That you can start getting an organizing matrix, wet-dry cycles. The polymers that are found are not equivalent to biological RNA, so we're not quite there yet, right? We haven't fully made the link from chemistry to biology, but we're getting there. Anyways, this is just more studies you can kind of check out. There's some assumptions that are still, again, it's It's not perfect. And yeah, there's still a lot of unsolved questions, right? What's the source of mononucleotides? How did RNA become capable of catalyzed growth and replication? How did biological homochirality emerge in the prebiotic racemic mixture? How did life begin to use light as an energy source? What were the first feedback loops? What were the first steps towards molecular evolution? How did complexes of RNA and peptides begin to function as ribosomes? How did genetic code begin to translate linear sequences? Anyways, now we do have prospects from life on other planets, right? You know Mars, hydrothermal fields like Columbia Hills, so what happened on Earth could have happened on Mars. Mars was in the Goldilocks zone for life, and you may have heard about some of the recent stuff that Curiosity and Perseverance rovers have found. Very exciting stuff. I know people that are on those papers, and I've published papers myself on some of this stuff, and Mars Guy's another cool YouTube channel that you can check out that talks about some of this stuff. And then there's other planets out there too, like Gillies 581, TRAPPIST-1, Kepler 90, that might also be habitable. You know, Europa and Enceladus in our solar system. There's a salt concentration underneath the ice of Europa and Enceladus, which we have actually studied with NASA probes. And they would inhibit self-assembly of membranous compartments. There's dilution of compounds with significant hydrothermal hurdles that prevent condensation. Alkaline pH promotes hydrolysis of ester and peptide bonds. There's no light, so again, we don't know if life could form kind of under the ice in Europa and Enceladus. It would be very different origin of life, maybe similar to the hydrothermal vent hypothesis. The warm little ponds hypothesis could work on Mars, could work on some other planets, but wouldn't work on Europa and Enceladus likely. Those are ice worlds that only have any sort of heat because of the gravitational interactions with Jupiter, but still could form. And there's thousands of galaxies. There's a lot of opportunities. The universe is too big for there not to be some sort of alien life out there. And you might think the odds are low, but when you have as many galaxies and opportunities that there are, you can definitely have life. So were hydrothermal ponds cradle of life? Could be. Again, hydrothermal field versus hydrothermal vents. And hydrothermal vents could be either white or black smokers as well. There's two sources of energy for hydrothermal vents: constant contact with seawater, which affects condensation reactions, evaporation, molecular crowding, and reduction in water activity are essential thermodynamic drivers for pre-polymer synthesis. And you need high ionic concentration. Vent mineral can catalyze the reduction of CO2 by dissolved hydrogen. These are some assumptions and conjectures. Substantial pH gradient forms between alkaline fluid and relatively acidic water. You have to have reduced carbon, can act as a substrate to initiate primitive metabolism, and pH gradients act as a source of chemiosmotic energy coupled to pyrophosphate bond synthesis. Some favorable conditions for hydrothermal fields: again, chemical energy from reduced water activities there, concentration on minerals, metal sulfides, self-assembly readily occurs in low ionic strengths, wet-dry cycles, which can increase complexity, we've seen this as well, and there's available phosphate minerals. So we need to better understand the, it's not in presentation, but you still see these things. We need a better understanding of the chemistry and physics of hydrothermal fields. We need a process that sufficiently concentrates diluted reactants, assembly of amphiphilic compounds into membranous compartments, a source of chemical energy to drive metabolism and polymerization, and a mechanism by which polymers can be encapsulated. We have some of these ideas, but obviously study work is still ongoing. And just a quote from Charles Darwin, right? Looking at the first dawn of life, when all organic beings, as we believe, presented the simplest structure, how it was asked could be the first steps in the achievement of differentiation. And again, abiogenesis and evolution are two completely separate processes. But anyways, all right, well, thank you for listening. Hope you enjoyed, and I will talk to you later. Peace.